Hello everybody, I'm Silas, and welcome to Psilocybology. Let's expand our mind. This is a series where we talk about the research on Psilocybin, the psychedelic prodrug found in magic mushrooms. We'll be breaking down and discussing the scientific literature, one study at a time, from past to present. If you'd like to support the show, please rate, review, and subscribe to the podcast wherever you can. And be sure to share the podcast with anyone you know who might enjoy the content. I really appreciate it. With the holidays upon us, I wanted to let you all know that there will not be an episode next Sunday, December 23rd, but that we'll be back Sunday, December 30th, to welcome in the new year with an exciting episode on the effects of psilocybin on color perception, just in time for those brilliant New Year's fireworks. Until then, I really hope everyone has been enjoying the holidays so far and continues to enjoy them as we make our way into 2019. I also wanted to give a quick thank you to my second iTunes reviewer. To answer the question that you left in the review, I do think an episode on interactions between antidepressant medications and psilocybin would be a great topic. I actually think that it would be great for that to be part of a series of episodes discussing interactions between psilocybin and different types of prescription medications. It might take a while to put those together, especially since I haven't yet come across research explicitly testing these drug interactions, but I can promise that that will definitely be something we cover. I can at least say that the researchers who examine the effects of psilocybin have stated that SSRIs and SNRIs will blunt the effects of psilocybin. I believe one reason this is the case is because psilocybin, and psychedelics more broadly, target the 5-HT2A receptor, which is a particular type of serotonin receptor. In response to treatment with some antidepressant drugs, such as an SSRI, there is downregulation of this 5-HT2A receptor, which means that the number of these receptors decrease. Essentially, by taking an SSRI, the goal is to increase the level of serotonin in the brain, which would then lead to increased activation of serotonin receptors. The brain, however, wants to maintain a particular level of activation, so in response to the increased serotonin, over time the number of serotonin receptors start to diminish, so that the overall level of activation remains the same. This is just one example of many homeostatic systems in our body, not incredibly dissimilar to how when it's hot we start to sweat, and when it's cold, we shiver. Our body is always trying to maintain a relatively stable body temperature based on a given set point. This is also why tolerance develops to some drugs, and a higher and higher dose is needed over time in order to achieve the same effect. The inverse process also exists, called upregulation, where in response to lower levels of a given neurotransmitter, the number of associated receptors increase. Anyway, thank you again for your review. Leaving reviews of the podcast really helps support the show, so please, if you're enjoying the podcast, don't forget to rate, review, and subscribe wherever you can. Getting into the topic for today, today's episode is on the article titled Effects of LSD-25, Psilocybin, and Psilocin on Temporal Lobe EEG Patterns and Learned Behavior in the Cat, published in 1962 by researchers from the University of California, Los Angeles, or UCLA. One thing I want to mention about this article before we jump in is that the primary focus for these researchers was LSD. Psilocybin and Psilocin, in this study, were really comparator drugs more than anything, so the authors spend much more time discussing LSD. There are also a few outcomes they measured after LSD administration that they didn't measure after psilocybin or psilocin administration. I figured that no one listening would be too upset by this. There's still plenty of psilocybin content to cover, of course. But I wanted to let you know up front that today we'll be giving a bit more attention to LSD. Also, for those who may not be familiar, EEG stands for electroencephalography, which is a measure of electrical activity in the brain and has most commonly been used in research into epilepsy and sleep. If you're familiar with sleep research, when researchers talk about the presence of beta, alpha, or delta waves as examples, those are all referring to the types of electrical activity recorded using an EEG. Now, I will point out that in humans, EEG recordings are typically taken from electrodes administered to the scalp and is thus non-invasive. However, in this study, they implanted electrodes in the brains of the 18 cats that were part of this experiment. This invasive form of EEG is also known as intracranial electroencephalography. So in this study, an EEG was used to examine how our drugs of interest affected electrical activity in the brain, and if any changes in electrical activity were correlated with changes in learned behavior. The researchers were particularly interested in the hippocampus and an adjacent region called the entorhinal cortex. These regions are critical for memory formation and consolidation, and particularly spatial memory. The researchers were also interested in the amygdala, primary visual cortex, subthalamus, and the midbrain reticular formation. Once the electrodes were implanted into these regions of the brain, the cats were administered either LSD, psilocybin, or psilocin via intraperitoneal injection, or injection through the abdominal cavity. Before getting into the EEG results, the researchers first describe general behavioral changes in response to drug administration. For LSD, they noticed consistent behavioral changes after administering between 25 and 100 micrograms of LSD per kilogram of body weight. To put that dose into a bit of perspective, if a 70 kilogram or 154 pound human being took the same weight-adjusted dose, that means they'd be taking, at the higher end, up to 7,000 micrograms of LSD, far surpassing what is often considered a heavy dose of about 300 micrograms. Just keep that in mind, and we'll revisit it in a little bit. So one of the behavioral changes they noticed was that about 30 minutes after being given LSD, the cats adopted what they termed a kangaroo posture, where they placed their weight on their back legs, which were also spread wide apart, and their tail was curved upward and their back arched. The cats also pawed at imaginary objects, possibly indicating visual hallucinations. They lied sprawled on the ground with paws and claws extended, and occasionally began shaking their head violently from side to side. The researchers also noted that the cats purred incessantly. I actually looked up if we know more about why cats purr. Personally, I really didn't. And I actually came across an article on WebMD that said there are a few different reasons cats purr, and that although we often think of it as meaning that they're happy and relaxed, it can actually mean much more than that. Apparently, cats also purr when they're in pain, as a potential way to soothe themselves, similar to a child sucking their thumb. Lastly, they noted that the cats engaged in less affectionate behavior, like rubbing up against the legs of observers. They also had goosebumps. Interestingly, they did not experience any pupillary dilation. Before we contrast this with the effects seen after psilocybin and psilocin, I want to make sure we keep in mind that differences seen are likely much more influenced by the differences in drug potency rather than the drug itself. What I mean is, the researchers administered between 0.25 and 1 milligram of psilocybin or psilocin per kilogram of body weight. If we were to use the same weight-adjusted dose in humans, a 70 kilogram or 154 pound individual would be given at the high end 70 milligrams of psilocybin or psilocin. This is absolutely still a very high dose. A heavy dose of psilocin, for example, is about 40 milligrams. However, a 70 milligram dose of psilocin is less than 2 times as much as what can be thought of as a heavy dose, whereas 7,000 micrograms of LSD is 23 times what's considered a heavy dose of LSD. It is clear that the researchers made absolutely no effort whatsoever to try and use comparable doses of LSD, psilocybin, and psilocin. As we've already seen from past episodes, it's hard for the researchers to plead ignorance, because although research up to this point hasn't definitively determined the relative potencies of these drugs, they have at least estimated the relative potencies with much greater accuracy than what they used in this paper. This is why understanding the research that's already been done is crucial when conducting new research. So that said, and keeping this difference in potency in mind, let's talk about the general behavioral changes seen after the cats were given psilocybin and psilocin. What they found was that the cats were very vegetative. They all experienced pupillary dilation, rapid respiration, vomiting, and an increase in urination. I want to note that the rapid respiration in particular is opposite to what we found in the previous episode, which found decreased respiration in the anesthetized dog. Although this might actually support the claim made by the authors of last week's paper that this decreased respiration they found was actually caused by an interaction between psilocybin and the anesthetic. Lastly, the authors note that affectionate behavior still persisted and that the strange posturing behavior was less common. They also noted that they observed some muscle spasms and skin twitching, although not the same violent head shaking. The next set of results get into the actual EEG recordings. After being given LSD, the researchers found that there were spontaneous seizures when the cats were in a dark and quiet environment. In fact, the researchers noted that the appearance of these seizures appeared to be dependent on the reduction of visual and auditory stimuli. The abnormal electrical activity disappeared when the cats were brought into a bright room or was required to participate in some of the learned behavior tasks, which we'll talk more about in a bit. The only exceptions to this were that there was an increase in seizure activity when the cats started eating food, and that at the highest range of doses, the seizures did persist somewhat even outside of the dark and quiet environment. As far as the brain regions impacted, these episodes of electrical activity were greatest in magnitude and longest in duration in the dorsal hippocampal and entorhinal cortical areas. They did also note the presence of this electrical activity, however, in the midbrain, thalamic nucleus, and primary visual cortex that appeared simultaneously with those originating in the hippocampus, but were of a shorter duration. They noted no or only minor seizure bursts originating in the amygdala. However, at the higher range of doses, they observed that seizure bursts that originated from the hippocampus appeared to propagate or spread to the amygdala. Comparatively, after cats were given psilocybin or psilocin, the researchers found a pronounced slowing of activity in the amygdala and greater high-amplitude slow waves in the visual cortex. Now, in the hippocampal system, slow waves were common both in the absence and presence of psilocybin or psilocin. However, after drug administration, the slow waves in the hippocampal system appeared to be more in sync with the presence of slow waves in other brain regions, where slow waves were normally less pronounced when the drug was not present. The last primary set of findings examined the effects of LSD, psilocybin, and psilocin on performance of learned behavior tasks, and if such performance changes correlated with EEG activity. The learned behaviors that the researchers observed were tests of memory. One such test was a delayed response task. The way the task appeared to have been set up was that cats were placed into a maze shaped like the letter T, and food was placed in one of the two arms or side branches of the T-shaped maze. The cats were given a brief visual cue telling them which arm of the T-maze had the food. The cue was then removed, and the cats were forced to wait between 5 to 10 seconds before they could move. They were then tested to see if they went down the correct arm of the maze that contained the food. What the researchers found was that when seizures occurred during the delay period, before the cats were allowed to approach the food, then there didn't appear to be any impairment in performance on the task, meaning they correctly approached where the food was hidden. However, when seizures occurred as the animals were moving towards the fork in the road, so to speak, where they had to decide which way to go in order to get the food, that's when the cats became disoriented and either failed to complete the task at all or made the incorrect choice. This was similar across all 3 drugs administered. Although that was all of the primary findings, they did conduct one additional test concerning LSD, specifically examining the development of drug tolerance that I just wanted to touch on. What the researchers observed was that when high doses of LSD were repeated between 3 to 7 days after an initial dose, no effect was seen either on the EEG recordings or from observing the cat's behavior. They didn't test this any further out than a week, so they can't claim how long it takes for tolerance to dissipate, but they can at least say that, in the cat, tolerance remained for at least a week. So in conclusion, the authors state that this research illustrates how LSD, psilocybin, and psilocin produced abnormal electrical activity that possibly originates from the hippocampal system, which then spreads to other subcortical regions. They also note that this is in addition to the changes produced in the primary visual cortex. The presence of these electrical disturbances appears more pronounced in the absence of visual or auditory stimuli. Well, that's it for today's episode. As always, I really hope you enjoyed the show. If you have any questions, feedback, or ideas for the podcast, please let me know. You can find out all the ways to reach me on the website, psilocybology.org, where you can also find the full transcript for each episode if interested. Thank you all for listening, and I'll talk to you next time.